Hi guys, in this lecture we're going to talk about DNA replication. And as you know, DNA, the molecule actually comes in the form of a double helix. It's made out of two strands that are twisted around each other. Unlike DNA, RNA just comes as a single strand. It kind of looks like a ribbon. It's not in the form of a helix at all or a helical shape at all. So it's a little bit easier to replicate, whereas DNA, a couple different things have to be done before we can actually start complementing different nucleotides to the original strand. First thing that has to be done is that it has to be unwound, and then it has to be unzipped, because those nucleotides on each side are bonded onto each other through, if you remember, hydrogen bonds that are pretty easily broken. So let's just kind of go through the process a little bit, as well as some terms and definitions. The first thing that I want to point out to you in this picture is this dark blue strand. There's two of them, okay? Right about here, the DNA is still in its, I wouldn't say helical form because it's not twisted, but it's still together, whereas those bonds, those hydrogen bonds that are in the middle here, holding all of these individual nucleotides together, will be broken. And basically what does it is this kind of light green molecule right here. That's actually an enzyme, which again is a protein, but that guy, helicase is its name, is actually going to unwind and unzip the DNA, and you can see that here it's kind of becoming unzipped. And then finally, the next part is, once these are unzipped, they're highly unstable. When they're together, they're very stable. When they're unzipped, they're very unstable. They need a stabilizer. So a couple different things happen. There's something called a topoisomerase. That's an enzyme that functions to basically relieve the strain that the DNA is being put under by being unwound. So right here, there's a lot of strain, okay? So there's going to be topoisomerase, another enzyme that's going to come in here and kind of help take to relieve that strain that's being put on the DNA. The next thing that's going to happen is you're going to get some molecules, again, some more proteins or enzymes that are called single-strand binding proteins. And that's going to help to basically stable these now two released strands of DNA. Kind of like a flag waving in pretty heavy wind. That's kind of what happens once these become unlocked from each other. So they definitely need to be stabilized some more. Alright, so a couple other terminologies here. These light blue strands that you see here, okay, those are the new DNA nucleotide strands that are being put on onto these dark blue strands. The dark blue strands are what we call template strands, or original or parental strands, if you will. I will always refer to them as template strands. And then the new strands of DNA, they also have a specific name to them. They're complementary strands. Now, right here, you can see right where the two strands are being pulled apart from each other, there kind of makes this little fork, okay? That's what we call a replication fork, okay? Now, if you look up here at this picture, this is usually what DNA looks like. Once the strands are pulled apart at some point in the molecule, because this whole thing is one molecule of DNA, and it could happen, replication could happen anywhere, they're saying that this right here is actually, right here, is the origin of replication. So that's where it all began. So this huge thing, once those are pulled apart, excuse me, that creates basically a replication bubble. Okay, and within that bubble, you have two ends here that are actually called forks, okay, or replication forks. You're always going to have two, unless one end is closed up completely, or at the end of the molecule. All right, but let's go back down here and actually look at what's going on here. So we've basically got these two strands apart. We're ready to start complementing. Now, if you remember, the nucleotide sequences for DNA, we have A, C, T, and G. So guanine, thymine, cytosine, and do you remember what A is? Adenine. So those are the four. And Now my next question for you is, do you remember which ones go with which? So which ones complement which? Well, it's pretty simple actually, but A's and T's will always go together, and C's and G's will always go together. And the reason being is, if you remember way back when we were studying these molecules, some of these are purines and some of these are pyrimidines, and it just has to do with some of these molecules, or these nucleotide bases, or nitrogenous bases, they are in the form of a double ring, whereas some of them are also in a form of a single ring. And as we talked about, you have to have, in order for the two ladder rims to be equally distanced apart from each other, you have to have a total of 3 rings, molecular rings across. So we couldn't have A to A, because we would be having, say, 2 and 2 rings together. So that'd be 4 across. So we couldn't do that. We couldn't have C and C together, or G and G together, because then we would have only 2 rings across, and we would have a ladder that kind of looks like this. Okay, so we'd have 1, 2, 3, 4, 1, 2, okay, and so on. You get the picture. So that's why this distance has to stay the same all the way across. So this is what you're looking for right here. A's and T's go together, and C's and G's go together. One way to remember is put these letters together and you get at. Okay, the other way that you can think of it too is that the C shape and the G shape are roughly the same. So that's another way to remember it if you're having troubles with that. But let's get back to what's going on down here. So basically the strands have been separated and they've been stabilized. So now this little guy right here, DNA polymerase 3, it is an enzyme. It's going to come in and it's going to start complementing this old template strand. Okay, so it's going to start reading it. Now one thing I'd like to kind of point out is, and if you're looking over here on the right side here, this 5' and 3'. Basically, all strands of DNA, I'm just going to draw one here. For instance, let's look at the top strand. So if you look at the original strand, it shows that you have a 5' on that side, meaning that this side is automatically going to be a 3' side. Okay, replication. So actually laying down the new DNA always goes from 5' to 3'. Okay. However, reading the old strand, it goes from 3' to 5'. That's going to be really hard to remember, so you're going to have to come up with a way to remember that on your own. But that 5' to 3', a good way to remember this is just think of opposites. As long as you know what one strand is, go opposite. So if this is the new strand, this has to be 5' because it has to be opposite of the template strand. Okay, so that means that this side would have to be 3'. So in what way are we actually laying it down? 5' to 3'. Okay, and we're reading 3' to 5'. Okay, so either way, we're still going in the direction of the replication fork. And that's for this strand, however. Okay, this is what we would call the leading strand. And one clue that gives you that this is the leading strand is that there are no gaps in this strand right now. It is a continuous replication of nucleotides. Whereas if you look down here, this strand has these little gaps. And you also notice these little red primers in there as well. And we'll get to that in a second. But this is what we call the lagging strand, because this strand takes a little bit longer to actually replicate than does this continuous replication of the leading strand. Regardless, overall, all replication always toward toward the fork. Okay. All right. So this DNA polymerase is going to start laying down new nucleotides, and it's going to read from 3' to 5', and it's going to start laying down new nucleotides from 5' to 3', and they have to complement each other. Okay, so A's and T's have to go together. So let's just say that we've got, an A down here. Okay, T up here. Well, if it reads that this is a T, then this would have to be an A, okay, and so on. If this is a G, then this would have to be a C, and so on, okay? So, and we'll go through how there are some ways that this whole process is able to proofread and repair and do things like that to make sure that there are no errors, okay? All right, so let's keep going. So this is basically going to keep laying down these nucleotides. All right, so great, that's the easy one. Now let's take a look at the lagging strand. The lagging strand is a little bit difficult, okay, because yes, overall it goes in this direction toward the replication fork, but it has to lay down going in this direction, and I'll tell you why. First thing, the reason being is the direction of the strand. Okay, we have our 5' over here and our 3' on the other side. So basically what happens is this primase guy right here, okay, primase actually lays down these RNA primers. These are literally RNA nucleotides that are going to complement the template strand just temporarily, okay. Basically what they're there for is to give DNA polymerase 3 a location to start making what we call Ozaki fragments. And what is an Ozaki fragment? If you take a look at this little patch of light blue right here, that little patch of DNA segment is what we call an Okazaki fragment. It's a fragment of DNA nucleotides that gets laid down by the DNA polymerase 3. So what happens? Okay, well, essentially what happens, remember the overall laydown is this way, but in order for DNA polymerase to work, it actually has to meet up with a primer and start right here. And it lays it down just like this arrow says. It actually lays down DNA nucleotides. Remember, the primer is made out of RNA nucleotides, but the DNA polymerase 3 is going to lay down DNA nucleotides, and it's going to do it starting here and going this way, which is kind of strange, right? So it starts here and it actually lays it down this way. Okay, so then what happens when it hits another primer? Well, that primer is there to say, hey, stop, we're good for right now. So DNA polymerase actually jumps from here and it goes to the next primer. And it starts laying down DNA nucleotides this way. And then when it's done here, it's going to continue over. And remember, by that time, this will have been opened up a lot more. Okay, so there'll be a lot more left to kind of keep going, and more primase will have laid down more primers so that the DNA polymerase will know where to start. So you can kind of see that the overall direction is this way because it keeps going this way, but when it actually lays down the DNA nucleotides, it lays it down in this direction. Okay, so what happens after these are laid down? Does the RNA stay there? No, it gets kicked out, essentially. So DNA polymerase 1 will actually come through, and here it is in this kind of yellow enzyme, okay? And as you can see, it's starting to remove these little primers, okay? So it's actually going to cut out the RNA nucleotides, okay? Kick them out of there. And then it's going to replace them with new DNA nucleotides. So it's actually going to proofread itself, and then they're going to throw in the new DNA nucleotides itself as well. So this is what DNA polymerase 1 is in charge of. It takes off the primer one by one, so one nucleotide at a time, reads that template strand of the nucleotides that are there so it knows how to complement it, and then it's going to stick in the new DNA nucleotides that go in there. Now, we're still going to get these little gaps, and you can see that there. Here's another gap. That's where DNA ligase comes in. It's another protein enzyme that comes in, and its job is really just to bind those gaps together. So it's going to take those DNA nucleotides, and it's going to bind them together, completing this strand. So it's going to connect the two Okazaki fragments here together. Now, what happens if the DNA gets screwed up somehow? Maybe it's misread or something, a nucleotide somehow gets bonded onto the old template strand and it shouldn't belong there, like maybe an A and a G for some reason get bonded together. Or there's some damaged DNA. What happens during that process? Well, these same types of enzymes are responsible for basically cutting out or incising, or during an excision repair. They're actually going to cut out that damaged area and replace it with correct nucleotides for that area. So how does this all start? Well, a thymine dimer actually distorts the DNA, kind of spreads it out a little bit so that the enzymes have a little bit more space to work and know exactly where the repair needs to be made. So it kind of distorts that area of the site of the damaged DNA. Then an enzyme called nuclease cuts out the damaged section of DNA. So let's just say that this is a damaged section right here. So it's actually, nuclease is going to come in, it's going to cut it out, okay, toss it away. And then DNA polymerase 1, right here, okay, kind of our true proofreader, is going to come in and is actually going to fill in that gap with the missing correct nucleotides. And then finally DNA ligase comes in and seals it all up again. So binds all that new DNA together to complete the strand. So these two guys play a very important role along with nuclease and a thymine dimer to help repair any damaged DNA. So thanks a lot. We'll cover some more at school. Thanks guys. Hope you're having a good break.